session, we'll talk about how to construct a first artificial RDNA molecule and who constructed it, right? So, our topic for today is construction of RDNA molecule and or who constructed the first artificial RDNA molecule. We can tell construction of the first artificial recombinant molecule. This we are going to see. Now, what is meant by recombinant? Recombinant means when there are DNA from two sources, that is called a recombinant DNA molecule, right? So, then who are the people behind construction of RDNA technology or biotechnology? Means always two scientists' name will always come in our mind. One is Cohen, other one is Boyer. So, again obviously these are the people who have first constructed the RDNA molecule. So, what are the names of the scientists? Stanley Cohen with Herbert Boyer. In which year did they construct the first artificial RDNA molecule? Means children, it is in the year 1972. In the year 1972, they constructed the first artificial RDNA molecule. I told you RDNA means recombinant DNA. Recombinant DNA means the DNA from two different sources. And we also know what are the contributions of Cohen and Boyer towards the biotechnology. We know that Cohen is a person who isolated plasmid from the bacteria and then he reinserted also. We know about the Boyer contribution also. He isolated restriction enzymes. Now, merging together, they were able to construct the first RDNA molecule in the year 1972. So, let us see that what they have done. So, Cohen, what did he do? Means from Salmonella typhimurium. From Salmonella typhimurium, they isolated a plasmid. This is the plasmid, which is called native plasmid, wild plasmid. They isolated it from Salmonella typhimurium. Now, into this, if there is only one DNA, then it's not called as an RDNA. We need to introduce a new DNA. Now, that DNA which we introduce is called as an alien DNA, right? So, the alien DNA, or I told you can also call it as desired DNA, or you can call it as gene of interest, right? Foreign DNA, all these things. Now, the alien DNA for Cohen and Boyer is antibiotic resistant gene. What is an alien DNA for Cohen and Boyer? So, it is an antibiotic resistant gene. This is the alien DNA for Cohen and Boyer. What is it? It is antibiotic resistant gene. Now, in NCERT, this much information is only given. The alien DNA or foreign DNA for Cohen and Boyer is antibiotic resistant gene. But if you look inside and if you see what is that name, what is the name of the antibiotic, means it is kanamycin. The name of the antibiotic is kanamycin. This is their alien DNA. Kanamycin, from which bacteria did they take then? They took kanamycin from another Salmonella bacteria which is called Salmonella panama. This kanamycin gene is taken from another bacteria which is called Salmonella panama. From Salmonella panama, they have taken the antibiotic resistant gene, which is their desired gene, and they are trying to insert this into the E. coli. They are trying to insert this into the E. coli. How can you insert this DNA into the E. coli? For inserting, you need an enzyme to cut it, right? So, then we know that Boyer has worked on a couple of restriction enzymes isolated from E. coli. So, they used, which enzyme they used? What is the restriction enzyme they used? To cut the DNA molecule. To cut the plasmid molecule means They used EcoR1. They used a restriction enzyme called EcoR1 to cut the plasmid DNA and the desired DNA. EcoR1 means it is a restriction enzyme isolated from E. coli. Isolated from a bacteria called E. coli, they have isolated a restriction enzyme. Now this DNA is able to cut that enzyme is able to cut the DNA in a precise manner. So we can call them as molecular scissors. Since it is cutting the DNA, we can call this E. coli, this EcoR1 or restriction enzyme as molecular scissors, or we can call them as molecular scalpels. Okay, so cutting devices are called molecular scissors. Now they used the EcoR1 enzyme to cut the DNA molecule. So they are digesting it now. When you digest this, what is this? This is a molecular scissor. It is a restriction enzyme. The name of the restriction enzyme is EcoR1. When you digest it, children, what will happen? It will open. In this manner, the plasmid DNA gets opened up. Now, in this region, now we are going to keep the desired DNA, antibiotic resistant gene, which is kanamycin gene. Now, we will give it here. Now, these ends should be sealed with the DNA. Now, what is that enzyme which is used for joining? It's called as DNA ligase. For joining, we use an enzyme called DNA ligase. For joining the fragment, we use an enzyme called DNA ligase. So, DNA ligase is called molecular glue. When this enzyme is called as cutting one is called as molecular scissor, then ligase will be called as molecular glue. It's called as molecular glues. Using the DNA ligases, molecular glues, we will join the antibiotic resistant gene with the plasmid DNA. Now, can I call this as a recombinant DNA now? Because there is a DNA from two different sources. Now, I got a recombinant DNA, which is called as a RDNA vector. You can also call it as chimeric DNA vector. For RDNA, we can also use a word called chimeric, chimeric. We can also call it as chimeric DNA vector. After getting the chimeric DNA vector, you need to send it into the host, right? So, they were able to send it into a bacteria. From where did they take the plasmid? They took the plasmid from Salmonella typhimurium. It is a native plasmid. Now, where are they trying to inject? They are trying to inject into another host, not the same host. They are not trying to inject back into the Salmonella typhimurium. They are trying to inject into another host. Its name is E. coli. It's a bacteria, E. coli, which is a relative of Salmonella. E. coli is a relative of Salmonella. How come you are telling that they are relatives of Salmonella? Means there is a family called Enterobacteriaceae. The name of the family is Enterobacteriaceae. All the bacteria which reside, entera means intestine. All the bacteria which resides in the intestine are called as Enterobacteriaceae members. All of us know that Salmonella typhimurium stays in the intestine only, small intestine only, right? So then E. coli is also an intestinal bacteria. Salmonella, E. coli, Shigella, Klebsiella, Enterobacter. These are few names I am giving which come under families Enterobacteriaceae. So then they are sisters to each other, they are relatives to each other. So they have taken the plasmid from Salmonella, they modified the plasmid and they are giving it to E. coli, which is relative of Salmonella. So, when they tried to send this recombinant DNA into the E. coli molecule, so E. coli, it took it nicely. E. coli has taken this recombinant vector. Not only that, so when this E. coli is multiplying, E. coli generation time is 20 minutes. Not only that, this E. coli, when it is making its progeny, E. coli when it is making its progeny, after 20 minutes, one E. coli cell will become two E. coli cells. Now, it is able to replicate the plasmid which you sent and the recombinant DNA, the gene marker what you have sent, antibiotic resistant, kanamycin what you have sent, is able to pass on to its progeny also. Means the gene what you have given is multiplying by using the host DNA polymerase. The plasmid what we have given has entered the new host. The recombinant DNA plasmid has entered the new host, and it can use the new host DNA polymerase enzyme to multiply and produce many number of copies. It can produce many number of copies, right? So, and it is passing on to its progeny cells also. This is what is RDNA technology. This is what Cohen and Boyer has done it. In which year? In the year 1972. Did you understand? Let's go once again. Today's topic is construction of RDNA molecule. The first artificial DNA molecule, RDNA, was constructed by Cohen and Boyer in the year 1972. For that, you need a vector. They have taken a plasmid vector from Salmonella typhimurium. It is a native plasmid. Then into this, you need to send a desired DNA. The desired DNA is antibiotic resistant gene, which codes for an antibiotic called kanamycin, which was taken from another species of Salmonella called Salmonella panama. Now, if you want to keep it this inside the plasmid, you need to digest this. For that, we require the help of restriction enzymes. The restriction enzyme what they have used for constructing the first RDNA is EcoR1, which is isolated from a bacteria called E. coli. It cuts the DNA in a precise manner. So that's why they are called as molecular scissors, or we can call them molecular scalpels. When you cut it, it gets opened up. So when you cut it, it gets opened up. When it opens up, in this place, you can bring this gene and you can insert it. When this has to get inserted, the end should be joined. Joining of this end is possible with the help of another enzyme, which is called as DNA ligase. Because it is joining, we can call it as a molecular glue. When you join both of them, you will get a recombinant DNA vector, which is also called chimeric DNA vector. Now, this recombinant DNA vector should be sent into the host. They tried to send it into another host, which is a close relative of Salmonella. They send it into the E. coli. So, E. coli took the vector, and this plasmid, which is inside the E. coli, is now able to use the E. coli's DNA polymerase, multiply and produce more number of copies, multiple number of copies it is making. This is construction of first artificial RDNA molecule. Take a screenshot. Right. Now, this is what is gene cloning, right? Now then, what are the three basic steps of genetically modifying an organism? We will see now. Hope you understood this concept. Now, the next topic which is there in NCERT is, what is that? The three basic steps of genetically modifying organisms, that we will see now. The three basic steps in making genetically modifying organisms. If you want to modify the genome of an organism, what are the three basic steps for that? The first one is isolation of the DNA with your desirable genes. We call isolation of the desirable DNA, alien DNA, foreign DNA, right? What's the first basic step? Isolation, identification of the DNA with the desirable genes. Okay, you brought the gene. Now you need to send it into the host. That's the second step. And see that the DNA what you have sent it into the host is getting maintained there, and it is producing and it is getting transferred to the progeny. These are the three basic steps. What are the three basic steps, children? The first one is identification of the DNA which is having the desirable genes. Sending that DNA into the host and then maintaining the DNA in the host and transferring the DNA into the progeny cells. These are the three basic steps. Let us write down. The three basic steps in making genetically modifying organisms are, first one is identification of DNA with You have to identify the DNA with desired genes. You have to isolate the DNA which are having desirable genes in it. Now next one, transferring the DNA into the host cell, right? So second one will be introduction of the DNA. You need to send the DNA into the host cell. Like a bacteria is a host, or plant can be a host, or animal can be a host. You need to send your DNA now into the host cell, because if you just keep it outside, it will not get multiplied. We want it in large scale, means you need to send it into the host, so then the host, whenever it is multiplying, your gene also gets multiplied, right? And the third one is maintaining the introduced DNA in the host, checking that the introduced DNA is intact in the host and checking that it is transferring it to the progeny, right? Then third one will be maintenance of introduced DNA. Maintenance of introduced DNA in the host. And transfer, see that transfer of the DNA to their progeny. See that the bacteria, whenever it is multiplying its progeny, that our DNA is also going into the progeny. Ensure that. Then only all the cells will be genetically modified. Understood? So these three. So in the previous lecture, we have seen what are the two basic principles of biotechnology: genetic engineering, chemical engineering. Now we have seen the three basic steps of genetically modifying an organism. Now let us talk about the next topic, which comes as the tools of RDNA technology. What is our next topic? Tools of RDNA technology. Let's go. Tools of RDNA technology, children. What are the requirements to do RDNA technology? All together will come under tools. Just now we have seen how Cohen and Boyer has done. For that, did they use restriction enzyme? Yes, they used. So restriction enzyme is the first tool then. Did they use the E. coli polymerase enzyme? Yes. Did they use ligase enzyme for joining? Yes. Did they take the vector from Salmonella typhimurium? Yes. Did they send it into a host called E. coli? Yes. So 5 things are important tools for constructing a RDNA. What are they? First one, for cutting, restriction enzyme. Then for multiplying that, we need polymerase enzyme. For joining both the DNAs, we need ligase enzyme. What are you making? How are you sending into the host? Through a vector. Plasmid is a vector. We need a vector. And into whom are you sending? A host. Like a plant is a host, animal is a host, bacteria is host. You need a host also. 5 things. Come on, let us list down. The 5 tools which we need for doing RDNA technology are: first one is restriction enzymes. Restriction endonucleases. It stands for restriction endonucleases. We need, right? The second one is DNA polymerase enzyme. The second one, what will this do? This will break the DNA, right? This will cleave the DNA, right? And what will this do? This will polymerize the DNA. And the second enzyme, the third enzyme is DNA ligase. What will DNA ligase do? It joins the DNA fragments, right? It joins the DNA fragments. The fourth one, we need a vector to carry our desired DNA till the host. So the fourth one will be vector. We need vector. What is vector? It acts as a carrier. Which acts as a carrier to carry the desired DNA to carry the desired DNA into the host cell. The last step will be the host. Now, what is a host? Host is the one in which the recombinant DNA gets multiplied. The host cell can be a bacterial cell also. It can be a plant cell also. It can be an animal cell also, in which we will introduce our recombinant DNA and ask the host to now multiply, now propagate it, right? So children, these are the 5 basic tools of RDNA technology: restriction enzymes, polymerases, ligases, vector, and the host cell. We will start with the first one, which is called as restriction endonucleases. Take a screenshot if you want. Right. Now I told we'll start with the first tool. What is that? Restriction enzyme. NCERT says restriction endonucleases, right? NCERT says in 1963, there are 2 scientists, the scientists' names are not mentioned in NCERT. In 1963, Lin and Arbor discovered 2 enzymes in a bacteria, they tell. Now, which bacteria? That also they did not mention. E. coli bacteria. So, in 1963, children, Lin and Arbor, they discovered that E. coli is able to protect itself from the bacteriophage. Now, we are infected by bacteria, virus, fungi, whereas bacteria is easily infected by a virus. Now, if the bacteria has to survive, then it has to protect itself from the viral attacks. So, what is this bacteria doing? How is it safeguarding from the viral attack? It seems the bacteria is safeguarding from the viral attack by a system which is called as restriction modification system. In that system, the bacteria is producing 2 enzymes. One enzyme is called methylase enzyme. One enzyme is called methylase enzyme and the second is called restriction endonuclease enzyme. Let us write down. So, in 1963, Lin and Arbor discovered that E. coli protects itself from the attack of bacteriophage, through a defense mechanism. It has a defense mechanism. Now children, this defense mechanism is called as restriction modification system. What is this defense mechanism called? It is called as restriction modification system. This restriction modification system, it produces 2 enzymes. The first enzyme is methylase and the second enzyme is restriction endonuclease. Now, this is the bacteria. If the virus comes, this is a viral DNA. Here is the bacterial DNA also. Now, it has produced 2 enzymes. One is methylase enzyme, other one is restriction endonuclease enzyme. Now, this restriction endonuclease enzyme, what it will do? When the viral DNA enters the bacteria, this restriction endonuclease cuts off the viral DNA. It cuts off the viral DNA into pieces. When the viral DNA is broken into pieces, then bacteria is safe now. Understood? So then this enzyme, restriction enzyme, it can also cut the bacterial DNA. So first its DNA should be kept safe. That means its DNA should be resistant to this enzyme. That is why first it will produce a methylase enzyme. What will this methylase do? It will methylate its DNA, own DNA. The methylase enzyme produced by the bacteria will methylate the bacterial DNA. Not every base gets methylated. Slowly in the next class you will come to know that the restriction enzymes cut the DNA at a specific sites called palindromic sites, which are called cleavage sites. Those cleavage sites where the enzyme can cut, there it gets methylated. When there it gets methylated, then this enzyme cannot cleave its DNA. It can cleave the non-methylated DNA, which is a viral DNA, right? So that's the action. Methylase, what it will do? It methylates bacterial DNA at the recognition sites, or we also call them as palindromic sequences. It will methylate the palindromic sequences, which is also called the recognition sequence for a restriction enzyme. It will methylate it so that the bacterial DNA is safe from the restriction endonuclease action. Then this restriction endonuclease, what it will do? The restriction endonuclease, it will cut the viral DNA. It will cut the viral DNA. That means the restriction endonucleases, which are synthesized by the bacteria, are they restricting the growth of the virus in the bacteria? Yes. That is why they are called as restriction endonucleases. They will ask you in your question papers, what they will ask? They will ask, what is the meaning of restriction in restriction endonuclease? The question is, what is the meaning of restriction? What is the meaning of restriction in restriction endonuclease? So what is the answer we are going to write? So the meaning of restriction in the restriction endonuclease is it restricts the growth, or it restricts the infection of virus in the bacteria. Answer is it restricts the multiplication. It restricts the multiplication of viral DNA or virus DNA in the bacteria. Understood, children? These two enzymes together will be called as restriction modification system. And this restriction modification system was studied in E. coli by Arbor and Lin in the year 1963. In that, if you want to do biotechnology, for that, will you use the former enzyme or will you use the later enzyme? What is the former enzyme? The former enzyme is methylase, whereas the later enzyme in the modification system, the later enzyme is restriction endonuclease. Restriction endonucleases are used for biotechnology. Restriction endonucleases are used in biotechnology. Understood? Now, we need to talk who isolated the first restriction endonuclease then. Now, if you want, take a screenshot, then we will see who is it scientist who isolated the first restriction enzyme and what is the name of it. Right? Now, the name of the other scientist is Smith. In the year 1968, he isolated the first restriction enzyme from a bacteria called Haemophilus influenzae. So, NCERT says that the first restriction enzyme to be isolated is HIND2. NCERT says the first restriction enzyme to be isolated is HIND2 from a bacteria called Haemophilus influenzae, which specifically reads a particular sequence which is of 6 base pairs and cuts the DNA in a precise manner. right? Let us see. So I'm telling, who is that scientist who isolated the first restriction endonuclease? It is Smith. In the year 1968, he isolated the first restriction endonuclease. I'm using a code REN for restriction endonuclease. Smith in the year 1968 isolated the first restriction endonuclease enzyme, and it is named as HIND2. It is named as HIND2 since it is isolated from a bacteria called Haemophilus influenzae. Since I told you in the previous classes also, restriction enzymes cannot be manufactured, cannot be synthesized artificially. You have to isolate it from prokaryotic organisms only. So, the first discovered restriction enzyme is HIND2 from a bacteria called Haemophilus influenzae, which identifies a particular sequence. It identifies a particular sequence of 6 base pairs. What is that 6 base pair sequence which is identified by restriction enzyme called as recognition sequence? What is it called? It is called recognition sequence. The recognition sequence for HIND2 also, children, they can ask us. NCERT says it's a 6 base pair sequence. Let us see the 6 bases also. It is GTC, GAC. What is the 6 base pair sequence? 5 prime it starts. It is GTC, GAC, 3 prime. The 6 base pair sequences GTC, GAC. Then base pairs we are telling. Then write the opposite strand also. C pairs with G, A pairs with T, G pairs with C, then C pairs with G, T pairs with A, and this pairs with C, then this is 3 prime. Now notice one beauty here. This type of sequences are called as palindromic sequences. The sequences which are identified by restriction enzymes are called palindromes. They are called as palindromes. So we'll write down the recognition sequences. The recognition sequences identified by restriction enzymes are palindromic sequences. What do you mean by a palindromic sequence? This is a palindromic sequence. Before looking into that, we'll try to understand in English what is palindrome. I'll give an example. You know, madam is a palindrome, right? Check this way, M-A-D-A-M, madam. Come from other way, M-A-D-A-M, madam only. One, a soap is there, Liril. I'll tell this is also palindrome, L-I-R-I-L. Come the other way, L-I-R-I-L, Liril. You can take Nitin also. N-I-T-I-N, N-I-T-I-N. NCERT has given an example of Malayalam. Ma-la-ya-lam. So, these are English palindromic words. Now, in DNA sequences, what are palindromic sequences means? This way you read, it will be the same in the opposite way. Now come on, GTC, GAC, right? So this way it is GTC, GAC. Now this way you read GTC, GAC. They are called palindromic sequences. All the restriction enzymes will identify palindromic sequences only. They will identify palindromic sequences only. For example, this is the palindromic sequence for HIND2, right? Let me take EcoR1 and show you. There's another restriction enzyme called EcoR1. There's another restriction enzyme called EcoR1. Now, what is a recognition sequence? Let us find out what is a palindromic sequence of EcoR1. It is 5 prime GAATTC 3 prime. Now, you are going to tell me in the opposite way what it will be. I told the one which is read in this manner, it will come in the opposite manner also. Means I have to write G, A, TTC. Now see, are they complementary? Yeah, G with C, A with T, A with T, T with A, T with A, C with G. Yes, it is 3 prime. So I showed you one more example which is called EcoR1 restriction enzyme. Its palindromic sequence, recognition sequence is GAA-TTC. And we are talking about the first restriction enzyme to be discovered is HIND2 from a bacteria called Haemophilus influenzae, which will identify a sequence which is of 6 base pairs, is called as a recognition sequence, and all the recognition sequences are palindromic sequences. So these are palindromic English words. These are palindromic DNA sequences of restriction enzymes. Now, what should restriction enzyme do? Just reading a sequencer, just identifying a palindromic sequencer, or cutting the DNA sequence also now. It's a nuclease. It has to cut the nucleic acid, right? So then the point where it cuts is called as cleavage site. Now, or cloning site. Where is the cleavage site? We will check out. The cleavage site for HIND2 is between C and G. Here then on that strand. And in the bottom strand also, where is CG? Here then. It cuts in this manner. Means it is cutting straight top and bottom. When such type of cuts are there, then you get blunt ends, I will tell. The DNA will become blunt. What do you mean by blunt? So because it digested here, this will be one part, that will be another part. Let us write. So GTC will come here. CAG will go here. And this way, 3 prime, 5 prime, CAG, CTG. This is called blunt end. I'll show you sticky end also. What are these end calls, ma? They are called blunt ends. They can also be called as flush ends. Blunt ends or flush ends. Now we took another enzyme also. What is that? EcoR1 is a recognition sequence. Now where is the cleavage site? Where is the cloning site? Here it is between G and A. It is between G and A here. Now in the bottom strand, where is GA? Here then. Yes. When you digest this manner, how do you get? So then your DNA will be like this. So it will be 5 prime G. It will be 5 prime G and it will be 3 prime. Let me draw it more clear. I'll use a black pen. Yeah, it will be 5 prime with G and 3 prime with CTTAA. CTTAA. The other strand will be AATTC. AATTC with this G. 3 prime, 5 prime. Now, how are these ends? See, these are blunt ends, right? Strict blunt ends. Such type of ends in this manner, I told you, they are overhanging 5 prime stretches in the first class, I think we discussed. These ends are called sticky ends. What are they called? Sticky ends. They are also called cohesive ends. Okay. They are called sticky ends, they are called cohesive ends, they are called staggered ends. They are also called staggered ends. So these are the names. Okay. So the first enzyme to be isolated by Smith in the year 1968 is HIND2 from a bacteria called Haemophilus influenzae, which identifies a 6 base pair sequence which is called GTC. GAC is its recognition sequence, and this site is called as cleavage site. This site is called as cleavage site or cloning site. And after it clones or cleaves, the resultant fragments are blunt ends or fleshy ends. If it is cutting between the on the top and bottom at the same place, you will get blunt ends. If it is cutting in this manner, you will get sticky ends. And the sequence what it is identifying is called as palindromic sequence. The meaning of palindrome is this way, it is the same. Understood? Now, this is our next topic what they have mentioned in NCERT. We are doing tools of RDNA technology. Under tools of RDNA technology, I told you enzymes like restriction enzymes, polymerases, ligases, vector and host are required. We started the first one, restriction enzymes, and then we came to know that like Lin and Arbor, they identified restriction endonuclease system. Then we came to know who isolated the first restriction enzyme. Now, how many restriction enzymes are there in the market is the next question. Now, 900 restriction enzymes are available, which are taken from, which are isolated from, 230 strains of bacteria. As I told you, we can't prepare it, you have to isolate it. So now how many are there in the market? 900 enzymes are there in the market, which have their own recognition sequences and cleavage sequences. Isolated from how many strains of bacteria? 230 strains of bacteria. Our next topic is nomenclature. You might be wondering, what is this EcoR1? What is this HIND2? We will see the nomenclature also in this session only. If you want, take a screenshot fast. Right. So one more point also I'll tell you. So I told about Arbor. I told about Smith. Another scientist is there. His name is Nathans. Now, he has also contribution in restriction enzymes. Arbor, Smith, Nathan, all three has contribution in restriction enzymes. Arbor, what did he do? He identified restriction modification system. Smith, what did he do? He isolated the first restriction enzyme. Nathan, what did he do? He used restriction enzymes in gene mapping. Nathan used restriction enzymes in gene mapping, locating the gene, he used it. So these three scientists, Arbor, Smith and Nathans, got Nobel Prize for the discovery of restriction enzymes. They shared the Nobel Prize in the year 1978. So what are the three scientists' name? Arbor is one person, Smith is another person with Nathan. They shared, what did they share? Nobel Prize. What? For their discovery of restriction enzymes. For the discovery of restriction enzymes. Now, let us talk about the nomenclature of restriction enzymes. Nomenclature, what do you understand by nomenclature? Naming, yes it is. Nomenclature of restriction enzymes. Now, we'll list down some names, right? Okay. Just now you saw I wrote EcoR1. One more you saw I wrote HIND2. Another one will be there, PVU1. Okay. So, like that, you have CLAV1, you have BAMH1, you have SAL1, you have PST1, you have like this. We have some enzymes names like this. Now, what did they name? How did they name? If you notice, in all these things, first letter is capital, right? So, the first alphabet denotes the genus name. It denotes the genus name. Means you should know that this enzyme is isolated from a genus called Escherichia. This enzyme is isolated from a genus called Haemophilus. This enzyme is isolated from a genus called Proteus. Then Caryophyllene, we will write down Bacillus, Streptomyces and Providentia, like that. So children, the first letter denotes the genus name of the bacteria. Genus name of bacteria, and written in capital letter, right? See there, and written in capital letter. It is written in capital letter. This is our first point. Next point. The next two alphabets are small letters. Next two alphabets. Next two alphabets are written in small letters. They are written in small and italics also. And italics also, and they denote, what do they denote? They denote the species name. Now, if the first alphabet is denoting the genus name, the next two alphabets, which are small case and which are mentioned in italics, will denote the species name. And if at all the fourth alphabet is there, in PST fourth alphabet is not there, here also fourth alphabet, here also it is not there, Hind it is there, Echo R1 it is there, BAM H1 it is there. So, the fourth alphabet, if present, then it denotes the strain, variety of bacteria is called strain. The fourth alphabet, if present, denotes the strain. So, it is not compulsory that you have to use capital letter only. It is not compulsory you have to use small letter. It is up to you. You can use capital letter also, you can use small letter also. Here we use capital, here we use small, here we use capital, like that. You can use either capital or small. Can be denoted either by capital letter, or small letter. Then afterwards, I am writing the Roman letters, right? 1, 1, 1, 1, 1, 2, like that. So, then the Roman number denotes, what is this Roman number? Roman number denotes the order of isolation from that particular bacteria. Today I isolated first enzyme from E. coli, then I can name it as Echo R1. Tomorrow if I isolate one more enzyme from the same E. coli, then I should name it as Echo R2. Day after tomorrow, if I remove the third enzyme, it will be called as Echo R3. Next day, Echo R4, like that. Understood? So, the Roman number denotes the order of isolation from the bacteria. Order of isolation from the bacteria. This is what is nomenclature of restriction enzymes. We'll go back again. The first alphabet denotes the genus. The next two alphabets denotes the species of the bacteria, italics, small letters. Fourth alphabet, if it is there, it is about the strain name. And then the Roman letter tells the order of isolation. We will take some names and we will see from which bacteria they have taken it. For example, Echo R1 says that it is isolated from Escherichia coli. E stands for Escherichia, which is the genus name. Next to CO stands for coli. We will write small letter because always the species should start with a small letter. So, coli, which is the species name. Then R stands for the strain. RY13 strain it is. It's a name. And 1 denotes the order of isolation. It's the first enzyme to be isolated from a bacteria called Escherichia coli. One more shall I show you? Shall we go with HIND2, which is the first restriction enzyme to be discovered? If we go with HIND2, now H stands for the genus name Haemophilus, right? And the next, second and third alphabets, IN stands for the species name Influenzae. This is the genus name, this is the species name, and the next alphabet D stands for RD, strain it is, and 2 is the order of isolation. You want one more? Shall we go with PVU? PVU2. P stands for the genus name. What is that? Proteus, which is the genus. VU stands for the species. Vulgaris. It is the species name. Fourth alphabet is not there. Then the 2 is order. It's extracted. PVU2 is extracted from a bacteria called Proteus vulgaris. Now, one thing you should get a doubt, like madam is telling the first enzyme to be discovered is Hind2. It should be Hind1, no? Why it became 2? It is like one person already took Hind1 and he is delaying his work. Meanwhile, the Hind2 work what Smith has taken, he did it fast. He did it fast, so that's why he could publish it fast. So that's why the first enzyme to be discovered is not Hind1. In fact, it is Hind2. Understood? So this information we saw here. In the next session also, we will discuss about the restriction enzymes only and their recognition sites and cleavage sites. Hope you are finding the lectures interesting. If you like the content, like, share and subscribe to my channel. Thank you.